Okay, so welcome to the seminar of the Australian Centre for Excellence in Antarctic Science. So today our speaker is Nicole Hill, who's a senior researcher at IMAS, and she's also a lead and a CI of ACEAS Program 2 on the marginal ice zone. So Nicole completed a PhD at UNSW, studying the ecological impacts of metal contaminated sediments on benthic communities. And she's then did a postdoctoral fellowship at NERP in marine biodiversity. For her current research, she uses statistical tools to quantify, protect, and monitor Southern Ocean marine biodiversity. So thank you, Nicole. I'll leave it to you.
Thanks, Laurie. Yes, today I'm going to be talking to you about some of the work that we're doing about mapping the distribution of Southern Ocean biodiversity and how it's changing. And I was going to give an introduction to myself, but Laurie did a great job, so I'll whiz through this really quickly. As Laurie said, I grew up in Sydney and I did my undergrad and my PhD at UNSW with Emma Johnson and the AAD and CSIRO Land and Water, looking at how the effects of metal contaminated sediments and a comparison between temperate and polar systems. And I was so lucky to be able to go to Antarctica a couple of times and have the most amazing experiences in the field doing my work with the most amazing people. And I think that's where I got bitten by the Antarctic bug, as I'm sure many of you here can relate to. And then I moved to Hobart to do a postdoc with the Marine Biodiversity Hub. And in this hub, we were really interested in using the biological data that we had to generate maps of the distribution of biodiversity. So we would start thinking, initially we started thinking about sort of single species, and then we started really thinking about, well, what is biodiversity and how do we quantify it more than just metrics of number of species? How do we capture all those complexities and how do we model what's actually there? And so that sort of piqued my interest in species distribution models and ecological models. And then that sort of information was fed into local and national marine park planning. And then once the Australian Marine Park Network was declared, the emphasis of the research sort of moved from this sort of predicting biodiversity to how then do we go out and monitor these enormous marine parks that cover this, they're remote, they cover large areas. We've got these maps of the predicted distribution of biodiversity, but when we actually have to monitor something, it's a little bit different knowing where to actually drop your gear to understand what's there and how it's changing. So using lots of remote technologies such as autonomous underwater vehicles, baited cameras, and some pretty fancy survey designs to get at an inventory and ultimately understanding how the systems were changing. And then I guess this has kind of led me full circle to working back to Antarctic systems and applying some of the modelling techniques and understanding that we developed in the Marine Biodiversity Hub to Antarctic systems. So as Laurie mentioned, my core research is really about quantifying, mapping and monitoring marine biodiversity with the ultimate aim of improving the evidence-based management of marine environments. So it's a common situation that managers require comprehensive spatial information, but the reality is that biological data is sparse and patchy. So how do we get from our discrete samples that look something like this across our area of interest to a map that managers can use in their decision-making frameworks? Well, one way to do that is to couple it with environmental data, and you can imagine that these coloured layers here are things like sea surface temperature, bottom currents, food availability, these type of things, with the biological measurements that we have, and develop these statistical relationships. And then once we develop those statistical relationships, we can essentially fill in the gaps where we don't have biological data and produce maps of what we think is there. And so this field of research is loosely based around species distribution models, but lots of species distribution models are interested in a single species at a time. when we're talking about biodiversity, we're actually interested in multiple species. So traditionally, as ecologists, how we would approach analysing multiple species is that we would start off with a matrix, something like this. We'd have a whole bunch of sites and we'd have a whole bunch of species and we'd record whether they are there or not. And this is clearly can be complex multivariate data. So in the traditional approaches that we would use, we would simplify this to a dissimilarity matrix. So pairwise, one number that summarises site by site, so pairwise combinations of how similar the species composition of these sites were. And then we'd run it through some kind of distance-based analysis. Lots of these routines are in a program called Primer. Now they're available in R. And then we would have to do some additional steps to understand and visualize what these analyses were telling us, because we've moved away from the actual species at sites to an abstract concept, a dissimilarity matrix. And then we need to, interpret that again to what we're actually interested in, which is usually which species respond to what environments and where are they. And then once we've got this abstract dissimilarity matrix, prediction is not necessarily that straightforward anymore. So there's been a recent suite of advances in the way that we approach analysing multivariate species data. And I'm calling these model-based approaches, multi-species model-based approaches, things like joint species distribution modelling, some names here, HMSC, regions of common profile, species archetypes. They're all sort of, particular techniques. But what they essentially have under the hood is that they're based on GLMs that are linked somehow, often through latent variable structures, which are things that we don't observe, but we model. So what this means is now, instead of turning things into a dissimilarity matrix, we're actually modeling the species at the sites directly. And we can get information directly from the model coefficients based on this. So, which species is responding in which way to environmental conditions, and how are they likely to occur over the range of particular environmental gradients. And this makes it much simpler to then turn this into maps about where we're filling in the gaps. So based on the environmental data we have at a site, how likely is it do we expect to see a particular species or group of species or assemblage at this location? And so this is an exciting new advance for ecological modelling. And so a couple of examples where we've done this in different contexts in the Southern Ocean space is an eco-regionalisation of the Kurgan Plateau. So as most of you probably know, the Kurgan Plateau is a significant feature in the southern Indian Ocean. It's a barrier to the Antarctic circumpolar current. It has a complex oceanography, which essentially results in a highly productive region that supports a diverse ecosystem from phytoplankton to seals and birds, and also commercial fisheries. So the northern part of the plateau, whoops, go back a bit. Where's the previous? So the northern part of the plateau is administered, it's a French territory and administered by France, and the southern part of the plateau is administered by Australia. And it's administered in accordance with the CAMLR's ecosystem-based approach to fisheries. In addition, in the southern Australian section, the Heard and McDonald Islands MPA was established in 2002, and in the French region, an extensive MPA was established in 2016. So every year in the Australian sector, and every probably 4 years in the French sector, trawl data is collected in this area, and that's primarily used to support the stock assessment for the toothfish and icefish fisheries. However, they collect information on all of the species that they collect, but to date, no one had really looked at this assemblage level data to understand what the distribution of assemblages were on the Kurgan Plateau. So this is exactly what we did, and we did this using a method called regions of common profile. And essentially what this method does is it uses this information on species occurrences at sites directly, and it tries to to find areas that have common profiles of species that can be defined by environmental characteristics. And you can interpret these as ecoregions or bioregions or assemblages, depending on the data and the scale that you're talking about. And when we did this for the Kerguelen Plateau, and I should back up a bit, we're talking about demersal fish here. Hopefully that was obvious. We found that we actually had 7 assemblages or 7 ecoregions that we could identify based on environmental variables such as sea surface temperature, bottom temperature, depth, productivity at the surface, and a range of other measures. And then when we mapped what the distribution of these assemblages look like across the plateau, this is what we get. So we get some shallower assemblages through to some deeper ones. And importantly, we can also quantify the uncertainty in our predictions. So we actually get predictions that are continuous, so probability of a particular group occurring there. And we can map how certain we are that it's going to be this particular group. So you can see for some of the shallower regions and some of the deep regions of the plateau here, we're pretty confident that it's actually this particular assemblage that we're looking at, whereas in some of the other areas we're a little bit less confident because it could be sort of overlapping between 2 types of assemblages. And this is also important to be able to quantify the uncertainty in the maps that we're giving people, giving managers to make decisions off. It's often something that's not particularly well done. So if we delve into what this yellow group is, so this ecoregion 7, directly from the model we can get this species profile. So these are all the species of fish that we modelled, and this is how likely they are to occur in that group. So this is again something that you can't get from the dissimilarity-based approaches directly. And we can from this information we can then say, well, what are the characteristic species of this particular assemblage or group? So in this case, it's the 2 ice fish, the toothfish, and the triangular rock cod are the most prevalent species in this group. And we can also, and one of the other neat things about this is because we've got this profile, so our average expectations plus a confidence interval, we can then plug new data into this and then see how good our models actually are. So we can conduct a sort of rigorous validation on this type of modelling approach. The other thing that we can get from it directly from the model is an indication of what the environmental conditions are at where these assemblages are found. So in this case, the yellow assemblage is a shallow assemblage. It's more likely to be found in shallow waters. It doesn't have a particularly distinct temperature profile, but it's also more likely to be found in regions with higher primary productivity at the surface. So once we've developed and mapped and understood the distribution of these assemblages, what can we then do with it? So it's a requirement for under the management of the MPA to assess its performance. So one of the questions that we asked with this information was how representative is the Herder Macdonald Island Reserve of the demersal fish assemblages? And so we basically calculated the overlap between each of the assemblages or ecoregions and the marine reserve. And essentially what we found was that there was reasonably good representation for most of the assemblages, except for this lovely brownie coloured one here, this assemblage 2. And when we dug into that a little bit more, we actually found that that was a fairly species poor assemblage dominated by toothfish. But this then gives managers more information on some of the representation gaps and what's in there and whether it's something that they should be concerned about and do something about or not. So moving on from that example, another example where we've used these new types of multi-species model-based approaches is looking at ecological change on the Kerguelen Plateau. So this is an Australian Antarctic Science Program, grant, and not to be confused with the AAPP, and it involves UTAS, obviously the AAD, CSIRO, and the University of Helsinki. And Joel Williams, whose picture is up here, he's the postdoc who is leading this program. So essentially, most of what we know about biological change in the Southern Ocean comes from a few key species. But we do have a few data sets where we've got good temporal information on whole assemblages. So, maybe not just penguins or a few species of seals, but we've actually got information on the full assemblage of organisms at a particular level. So one example of this is the fish trawls that I was talking about. As I said, they were conducted every year. So we have about from 1997. So we have, more than 20 years information of time series information. And another good long-term data set that we have is the continuous plankton recorder, which obviously collects zooplankton data for across the all across the Southern Ocean, but it has a high density across the East Antarctic section. So that allows us to start asking questions like, apart from how have a few key species changed, asking if, how, and why whole assemblages of species have changed in recent years, and in response to what, and if they have changed, then can we say, what are they likely to look like in the future? And so this is work that's underway at the moment. And the way that we've approached this is a joint species distribution modelling approach called hierarchical modelling of species assemblages, HMSC. So essentially what this approach does is, rather than modelling each species completely separately, one by one, it models them all together and it links their occurrences through these latent variables, which can model spatial and temporal structures. So basically, it gives a correlation structure to the co-occurrences in the data. And the advantage of doing something like this is that it helps rarer species be modelled better, and it can give us more accurate distributions than if we did single species individually. So in this situation, Joel's modelled the most prevalent 35 species. And what we've got here is a plot on, whoops, back. This plot over here, we've modelled the occurrence of these 35 species. And this plot over here tells us how important the environmental variables that we've put in there, plus some random variables to do with the sampling, are in determining their distribution. So we can get this for individual species, and we can also produce summaries for the entire assemblage. So in this graph here, you can see that this light blue bar is quite important for lots of species, and that's depth. So that's not surprising. Lots of species have a defined depth range. And some of the other important variables that are coming out for determining the distribution of several species are things like the southern annular mode, and so on and so on. So we can understand at the species level and at the assemblage level what are some of the environmental drivers that are important in determining their distribution. And then we can also look more closely at the response of particular species to the environment. So I've just picked out a couple of species here and a couple of environmental factors. So if we look at Patagonian toothfish, we can see that they're likely to occur all across the plateau here. They don't have any real preference for temperature, and they haven't increased over the years. They've remained fairly constant. If we have a look at the mackerel icefish, you can see maybe a little bit of a trend. So this is the temperature anomaly, but there's not much there. But if we look at the grey rock cod, you can see that it seems to like warmer, the warmer temperatures. And if you look at what's happened across the years, you can also see that icefish have become more prevalent, and so has the grey rock cod. So we can use this to understand how particular species are responding to their environment and through time. And the other neat thing that we can do is that we can then map this spatially through time. So this is really hot off the press sort of preliminary results here. But if we look at the Patagonian toothfish, we can see some it's as the model suggested before, it's highly prevalent across the plateau, but we can see some areas where we get more red through time than others. With ice fish, you can really see that trend for increasing prevalence through time, as you can with the grey rock cod. And then if we combine all this information from the individual species, we can also look at species richness through time. So a kind of community or index or metric. And we can see that there seems to have been a trend for increased prevalence of species through time. So now Joel has also modelled the abundance of these particular fish through time, but that's still being finalised, so I'm not going to present that today. And we're also able to consider the influence of species traits on their environmental response and distribution within this framework. So we can include things like whether they're fished or not, whether they're slow growing, large bodied, maximum size, all sorts of things we can include and see if there's any patterns there that relate to environmental factors. And within this project, we'll also be undertaking some future projections based on future scenarios. So what I want to talk about next is a large ARC project that we're involved in that's looking at mapping and predicting benthic biodiversity on the Antarctic continental shelf. So it's applying some of these techniques that I was just talking about in a different context to a different ecosystem. So this is a large project that involves obviously us, the Antarctic Division, British Antarctic Survey, but a whole bunch of collaborators from around the world who've contributed data and expertise. So essentially, the aims of this project in a nutshell are to provide fine-scale circumpolar maps of benthic biodiversity to provide a step change in the information available for spatial management. And there are a number of elements to this, as the diagram indicates. We need to generate some of the input data that we then use to model with these multi-species approaches. And then we're intending to produce a range of outputs or mapped outputs that relate to biodiversity. So distribution of species and eco-regionalisation, like I just talked about, and delve into some other key and topical issues for Camellara in the Southern Ocean at the moment, such as vulnerable marine systems and blue carbon. So what I'll do now is I'll just touch on where we're at with a couple of these sections to give you a flavour for what the project is about. So initially we needed to generate the data, the biological data to work with. So this is actually a huge undertaking, pretty much done by Jan Janssen, Victor Shemelov and Charlie Gros with the aid of Thomas Windsor. So we collated all of the available benthic imagery from around Antarctica. We utilised the downward facing imagery because it gives us the most consistent imagery between different surveys. And then we annotated that data in a consistent manner. So lots of this data had already been annotated, but by different groups for different things, using different schemes and different approaches. So it wasn't readily comparable. So what we've got here is a consistently annotated circumpolar, rigorously QA/QC data set of benthic biodiversity. So we have image data from 200 to 1500 metres depth, from 19 cruises across 286 transects. We scored about 3,500 images, and we have hundreds of thousands of points where we've detected substrate and fauna, and VME taxa. And I can't emphasize how much of a collaboration this was with international partners to get this data to us, and how much effort it was to actually score this information. But now that we have this data, it's an incredible data set that we can use not only in this project, but can be reused in other projects for different purposes. So the second component that we were looking at in this project is developing quantitative metrics of food availability at the seafloor. So because we're using models that require environmental data, it really helps to have environmental data that you think is directly determining the distribution of species. So depth is one that's commonly used, and it works really well, but depth is probably not the direct driver of a species distribution. It's probably other things like attenuation of food, pressure, light, that kind of thing. So one of the key things that we were missing until very recently is the distribution of food available to seafloor fauna. Obviously, this is going to be something that's important in directly determining where they are. And so in his PhD, Jan Janssen developed a method to quantify the food available at the seafloor using this neat combined modelling approach, where essentially we used the input from satellites. So we used satellites to quantify chlorophyll A, or primary production, at the surface, then coupled that to an oceanographic model. So I used the satellite data to seed the oceanographic model, transported these particles through the various layers of the oceanographic model until they reached the sea floor, and then looked at where they initially sank, where they moved to, and where they finally settled based on seafloor currents. And so Jan did a proof of concept of this for George V shelf, and it worked really well, and we found that it improved our ability to predict where sessile species lived significantly. And so in this project, we're expanding that to a circumpolar scale and a 4-kilometre resolution. So the oceanographic model that was under the hood for this was the ROMS system. And in this, once we're expanding it circumpolar, we're also updating some of the sedimentation and resuspension processes that are occurring at the seafloor. And this is work that's happening in collaboration with Ben Gouten Fenzi, and also tracking sources and sinks of primary production. So what you can see on the left here is from the ROMS model, and it shows the current speeds, absolute current speeds at the seafloor. And obviously this is going to be important in redistributing seafloor food. And these results are also hot off the press. And then on the right-hand side, you can see the sedimentation after 1 month of running the model. So the model uses climatological averages of sea surface primary productivity to flow through the system, and this is after 1 month where it ends. So this gives us information of sort of where some of the concentration points are for where we get more food at the seafloor and less. So this is something that we'll be using in our species distribution models. So that's actually really neat to see it at the circumpolar scale and at such fine resolution. And where Jan's in the process of interpreting these results and validating them at the moment. We validate them against sediment cores and the superficial layer of sediment cores, so either diatom abundances or total organic carbon is what we're validating them against. And the other neat thing that we can do is start to look at sources and sinks. So as I said a couple of slides ago, there's traces in the system. So this is focusing in on the region near the Ross Sea ice shelf. And you can see here, this is a figure of the traces where they are after 1 month. So there's no primary productivity under the ice shelf, but you can see after 1 month, their currents start to entrain some of the particles under the ice shelf. And if we go to 4 months, you can see some patterns where you can see that there's sort of a flux out from under the ice shelf, but there's also entrainment of particles under the ice shelf. Now this is really neat because it's not something we've been really able to observe before, and it'd be really nice to actually know what's under there. But there is precedence for this. So recently there were sessile filter feeding assemblages found 260 kilometres from the front of the Filchner-Rhone ice shelf around here, around there, when they drilled some holes for other purposes. So this is really neat stuff. So stay tuned. It's really hot off the press. So one of the other outputs that we want to generate from this work is an eco-regionalisation. So CAMLR is committed to establishing comprehensive and representative networks of MPAs. Some are established, many are proposed. But sparse biological data means that most of the bioregions that currently exist, like this one over here, it was published in 2014, rely heavily on environmental data as surrogates for biodiversity. Some of them include expert opinion and ad hoc biological data. But the process that we're proposing explicitly combines biological with environmental data and essentially puts the eco back into eco-regionalisation. It's also a process that is transparent and repeatable. So if we get more information, more biological information, different environmental information, we can rerun this process pretty easily. So the approach that we're using to do this, and we're running these analyses at the moment, is the one that I showed you from the Kerguelen Plateau, where we're looking at regions of common profile to understand where the distribution of the ecoregions are, what they're comprised of, and what the environmental conditions are like. You know, and for example, this is just a hypothetical, we might find that, you know, one particular bioregion over here has these amazing glass sponges. We might find another one over here has these dense assemblages of filter feeding organisms. Well, another one over here has is more dominated by sediment fauna. And the relevance of this, as I alluded to, is that we're explicitly putting the bio or the ecology back into bioregions, and we can evaluate the current and the proposed network in terms of representativeness. We can look at a gap analysis. So if things aren't covered in the current network, where would the most bang for buck be to extend if there was will to do that? And we've got a transparent and repeatable framework when if more information comes online. So one of the other aspects of this project is looking at vulnerable marine ecosystems. So VMEs, they're commonly called. Lots of taxa, lots of Antarctic taxa are long-lived, slow-growing, and low productivity. And collectively, organisms like this that are vulnerable to damage, particularly from fishing, but potentially from other threats, collectively form a vulnerable marine ecosystem. And the UN General Assembly had a resolution in, I think it was 2006, that really called on regional fisheries management authorities to have a precautionary and ecosystem approach to sustainably manage their fisheries with specific reference to impacts on benthic systems. And so CAMLR's response is to attempt to avoid significant adverse impacts on VMEs, which is, you know, an admirable thing, but in reality, the distribution of VMEs is poorly known, and often activities are only restricted after VMEs have been detected and at a particular threshold. So there are rules that relate to how much of particular VME indicator taxa hauled up on the fishing lines, whether that determines a move on rule or a spatial closure for an indefinite amount of time. So there have been an increase in recent years in studies that attempt to use species distribution modelling techniques to model the distribution of VME taxa. Often they only focus on a key taxa or indicator species, but VME is an ecosystem, as the name suggests. It's defined by several criteria: its uniqueness, its fragility, its functional significance, and more easily to quantify the life history traits. So this particular body of work is a body of work that Charlie Roy is doing as part of his PhD, and he's developing an index to consider the whole assemblage and multiple aspects of vulnerability to a particular threat. So rather than just thinking about modelling one particular species because it is an indicated taxa or it has particular life history traits or it is of structural significance, he's trying to encapsulate those characteristics, the vulnerability of the entire assemblage. And this work follows on from some work that's been done in the North Atlantic by Morato. So essentially what he's doing is incorporating the abundance of each particular taxa, so VME taxa, modulating it with a vulnerability score that considers these things: uniqueness, fragility, functional significance, and life history traits, to then come up with a composite index initially for each species and then for the whole assemblage about the vulnerability of that assemblage. So at the moment he's doing, and this index will be tailored for the Southern Ocean taxa and the CAMELAR criteria, which are similar to but different to some of the other regional fisheries management authorities. So at the moment he's doing this using a proof of concept using the images that we've scored from our database. And once he's completed this, he'll apply this index to the predicted distributions of species around Antarctica. So he'll be producing maps of the distribution of individual VME taxa and an integrative vulnerability index. And the relevance of this work is that again, we can look at how well current spatial protection measures are representing these hotspots and identify any gaps. We can look at vulnerability to different threats. So currently we're focusing on fishing, but we could potentially focus on vulnerability to climate change, for example. And in this case, the risk can be assessed before the activities take place. So if we know that there's a whole lot of particular areas that are likely to contain VME taxa, we might have different rules about what you can do in there, or different thresholds about when you have to move on and these kind of things. So another component of this project is looking at the blue carbon at the seafloor. So recently there's been recognition of the importance of high latitude shelves to the global carbon system and the role of benthic organisms in that. So essentially there's a range of ways that organisms can capture organic carbon, the CO2 from the atmosphere, and on various timescales. And when we're talking about the benthic fauna, we're really talking about this sort of immobilization, sort of years to centuries. So it moves through the food chain down to the benthos. It gets absorbed in the skeletons of benthic organisms, where it gets held for a long time, or it can get even buried, or when these organisms die. And recent work has shown that it can provide a negative feedback to climate change. And so this is really the seminal work of David Barnes at BAS, and he's going to be heavily involved in this component of the work. So we'll be building on his expertise and knowledge to then convert biomass estimates for the functional groups that we have to carbon storage. So this will be based on specimens and empirical data that David Barnes has collected from a whole range of benthic organisms to develop maps of carbon immobilisation at seafloor. So basically, we'll be using the empirical measurements that he's generated for functional groups, applying them to the distribution of these functional groups and generating estimates of the blue carbon immobilisation potential. And the relevance to policy of this is an increased importance of this ecosystem service and potential options for protection, which have been generating quite a lot of interest in the literature lately. And obviously an assessment of the carbon storage potential for the Antarctic benthic shelf. So from this project, collectively, the outputs that we're anticipating to generate, we've already generated the comprehensive compilation of the downward facing imagery and a unique data set of consistently scored benthic images, which once we publish our first paper will be made publicly available for people to reuse. Fine scale circumpolar maps of food availability at the seafloor, and then fine scale maps of the distribution of fauna, VME, bioregions and carbon. And this will generate an improved evidence base for management and conservation and policy decisions through, firstly, an assessment of current and proposed MPAs, a more precautionary approach to VME protection, quantification of benthic shelf carbon storage, and all of this within a transparent and repeatable framework that can be redone, revisited as more information comes online. So off the back of this project, where we transition into what we'd like to do with, or what we're going to do within ACEAS in program 2, is to essentially link what we're generating from this project to the oceanographic, the improved oceanographic and Earth system models that are being developed in ACEAS to understand what the future of benthic biodiversity around Antarctica may be. So we'll be revisiting these food availability models. So this will also be with Ben Galton-Fenzi and the models he's developing in that framework. We're very interested in other environmental predictor variables, particularly future projections of what these things might look like, such as sea ice and temperature and all sorts of other relevant variables. And then use this new information with the models that we've developed to sort of forecast or project into the future what the distribution of these various aspects of biodiversity, benthic biodiversity might be. And obviously the end goal, provide this information to policymakers to help them inform what they should do now and into the future for the Southern Ocean. And the postdoc on this, Jan Janssen, is expected to begin March next year. And one sort of final, so, the time frames that we're looking at, sort of 2050 to 2100 is where we'd like to be projecting into the future. And once we have some key people on board, there's some potential related PhD projects to this project. One of them is looking at a little bit more at this benthic pelagic coupling, where we have good data and oceanographic models and in situ chemical data as well, biogeochemical data. And the other one is related to another gap that we have in our understanding of environmental factors. So current and future distributions of icebergs. So there's interest in that from a number of aspects. So one, how it affects the benthic biota, but it's also important for understanding meltwater and iron fertilization rates, and also understanding past and present iceberg scouring and how that's affected the geology of the region. So thank you.
Thank you so much, Nicole, for a great talk. Does anybody have a question for Nicole? Okay, so while people think or even write in the chat, I have a quick question, but maybe I didn't understand properly. So you did mention temperature, so you were taking into account temperature in your modeling or in your statistical modeling. You never mentioned ocean acidification, and I guess if you're going to project for the end of the century, that could be important, no?
That's true. So in future scenarios where we have that information, then we can use that with what we know about the composition of the fauna that we're interested in. So we don't have that information right now, but if we have good fine scale predictions, we can use that for the future projections.
Okay, thank you. Cara Miller, please.
Hi, thanks very much for your presentation, Nicole. It's really interesting to see all your work laid out like that. I had a quick question about your regions of common profile. I was just wondering if there was a possibility of integrating age classes within that. So perhaps putting, juveniles versus adults for a particular species.
Yeah, absolutely. So we could treat them as we can break up the life histories and treat them as species or subspecies and get a finer picture of what that distribution looks like. Yeah.
Yeah, I know it wouldn't be possible for every species, but some there's age information, so that would be really interesting to look at as well.
Yeah, absolutely. Happy to talk more about that.
Yeah, thanks.
Matt.
Thanks, Nicole. And excuse my limited vocabulary, and I'll stumble over the concepts here. But I was interested in when you were talking about the Kerguelen work, and I started thinking about food availability then, and then you talked about that a bit later on. But I'm just wondering how complex the models are in terms of the environmental forcings on the food sources themselves, and I guess that cascades right through the food web at that point. So how do you deal with that?
So for the food availability modelling, or for the
so when you're looking at environmental forcings on a species, then presumably that's affected by the food availability. The food availability itself is presumably affected by different environmental forcings at times, and so on. Am I making sense?
I think you're getting at food web interactions. Is that what you're asking?
Yes.
Yes.
Probably.
Okay. So this approach is really a correlative modelling approach. So it doesn't directly deal with mechanistic what the mechanistic modelling approaches do, which is this particular species eats so much of this species, and this is a rate limiting factor on that. So it doesn't do that kind of modelling, but what it's doing is trying to get more of a distributional picture of why things are where. So if we have information on the distribution of food, then absolutely we can put that in, which is what we tried to do for the seafloor. There's some interesting work happening at the moment looking at the distribution of mesopelagics, which obviously, and zooplankton and so on. Obviously, if we had information about their distribution, we could put those into the model too and look at how that relates. Does that answer your question?
Yeah, I think so. I mean, I was just wondering about, once you start thinking about climate change and different forcings, and it becomes, obviously very complicated. And I'm just wondering where the limit of your modelling is when you're thinking about future climate.
Yeah, so that's a good question. So I think the correlative models get us a certain way, and I think they're well complemented by some of the mechanistic modelling approaches. They have different assumptions and data requirements. One of the nicer things about this type of modelling is that we don't need to know as much about the mechanistics of every species that we're modelling. And so I think they provide nice complementary approaches that tell us about different aspects of how things might change in response to future climate.
Great, thanks.
So there's a question in the chat from the ANU seminar room, which says, great talk, Nicole. It looked like there was a gap in the benthic maps in East Antarctica. Is there a plan to get images from there, especially near the Australian bases?
Yes, well, we are using downward-facing imagery because it's the most consistent across all of the different platforms that are used by different countries. There's not much imagery in East Antarctica, and there's not much downward-facing imagery in East Antarctica. Some of what we have is much shallower than what we're looking at. So we chose to look at sort of 200 meters and deeper because, you know, a lot of the really shallow water is influenced by, you know, fast ice and iceberg scour and a whole bunch of other things that we aren't quantifying well in this component project. So we chose to look at 200 meters and further down. There are a couple of voyages coming up that might help us fill those gaps, but it depends on what camera systems they use, and we will, given the time frame of this, we will likely use that as a validation data set for the models that we're producing, so we can tell how good the models are that we're producing based on these new data streams that come available maybe next year or the year after.
Thank you. And one final question from Rob Masson, saying, thank you for a fascinating talk. Just to comment that icebergs may also affect ocean currents, particularly when grounded in waters shallower than about 450 meters.
Mm-hmm. Yep.
Yeah, I'll comment.
Getting a handle on that would be great.
Okay, so anybody else's question or?
Yeah, I'll ask a question.
Oh.
So yeah, I guess I was just wondering, can we say anything yet about like whether there's large scale ecosystem change on the Antarctic continental shelf? And if we can't say anything yet, is that because there's not enough data, or can you robustly say that there's no change yet?
So for the system, for the benthic system, I think there's evidence in some regions of change. So particularly where, so a lot of David Barnes's work has seen, you know, when ice shelves collapse and whatever else, you know, you get that opening up of free water essentially in these intense plankton blooms, and you know, if that happens year after year, then you get a change, get a reasonably quick change in the fauna that occurs under there. So you end up with either more sessile, early successional sessile filter feeders, or more mobile, you know, deposit feeders that come in and then colonize those areas. So I think there's you know, regional scale evidence of these changes, but more broadly, I think, you know, that's what we'd like to get to. Yeah.
Cool, thanks.
Well, thanks a lot, Nicole, for a fascinating talk. I think we'll keep at this, but of course, people are free to contact you with more questions. I also make an announcement for next seminar, which will be a hybrid event. So Rebecca McGeer will give a seminar in person at ANU, and this should be streamed live into IMAS, as well as the CCRC seminar room at UNSW. It will also be, of course, available through Zoom as normally. Well, thanks a lot to everybody, and we'll see you next month. Bye.
Thanks, Laurie.